Hi, this is Edward Awad, and welcome to this video about biological diversity part 2. In the first video, Biological Diversity part 1, we looked at viruses and how eukaryotic cells are thought to have first appeared on Earth through the process of endosymbiosis. In this second video about biological diversity, we will look at the big lineages of living organisms and the principal evolutionary trends that shaped each of these lineages. All organisms are believed to have a single common ancestry. Through evolution, Earth became populated by genetically independent kinds of organisms that we call species. Speciation, which is the splitting of organisms into separate species, has resulted in the great diversity of life. It is estimated that more than 30 million species of living organisms inhabit our planet. Species are grouped according to their evolutionary relationships. Biologists now categorize all life forms into three domains: bacteria, archaea, and eukarya. This categorization is mainly based on the comparisons of nucleic acid sequences between species. The domains archaea and bacteria are believed to have first appeared some 3.6 to 3.5 billion years ago. They consist of unicellular organisms and are all prokaryotic. The third major branch, the domain eukarya, appeared later on in the evolution of life on Earth, approximately 2 billion years ago. Eukarya consists of a wide range of strictly eukaryotic organisms, from single-celled organisms seen in the lineage Protista to complex multicellular organisms such as plants and animals. The largest and most morphologically complex organisms of the tree of life are eukaryotes. Looking at this simplified tree of life, we notice that, from an evolutionary point of view, archaea are more closely related to eukarya than to bacteria. The domain eukarya is divided into four major lineages that traditionally used to be called kingdoms. But the word lineage is a more acceptable terminology and is widely used by biologists nowadays. The lineages of eukarya are Protista, fungi, animals, and plants. As you can see in this tree of life, almost all eukaryotes' lineages are considered as protists, except animals, fungi, and plants. When we compare the genetic makeup of the three domains, we can make the following general observations. First, the ribosomes of bacteria and archaea are structurally different from those of eukarya, and in general smaller than the eukaryotic ribosomes. Second, the shape of prokaryotic chromosomes is circular for most species, whereas eukaryotic cells have linear chromosomes. Eukaryotic genomes are in general much bigger than those of prokaryotic species, while in prokaryotes, a haploid genome has an average size between 1 and 5 million base pairs. In eukaryotes, the range is between 10 and 3,000 million base pairs. Third, the large size of eukaryotic DNA seems to necessitate its proper packaging in the nucleus, thus requiring histone proteins which serve this packaging purpose. Fourth, when compared to bacteria, archaea share more with eukarya similar gene expression machinery and molecules. And finally, only prokaryotes possess plasmids, which are extra chromosomal DNA of small size, usually a few thousand base pairs. When we compare the cellular characteristics of the three domains, we can make the following general observations. First, only eukaryotic cells possess a nuclear envelope consisting of a double phospholipid-based membrane. Second, prokaryotes lack any membrane-bounded organelles, while eukaryotic cells have extensive network of membrane-enclosed organelles of different kinds. Third, the structure of the plasma membrane lipids is unique in archaea and consists of glycerol that is bonded via ether linkages to branched short chains of fatty acids. The structure of the plasma membrane lipids in bacteria and eukarya is quite similar and consists of glycerol bonded to unbranched or straight fatty acids via ester linkages. Fourth, cell walls materials differ dramatically between domains. In bacteria, the cell wall is present in most species and consist of peptidoglycan, which is a complex polymer formed between modified carbohydrates and peptides. The cell walls of archaea come in variable structures and chemically are different from bacterial cell walls. In eukaryotes, cell walls are present in certain groups of lineages. Cells of plants and photosynthetic protists possess cellulose cell walls, whereas fungal cell walls and certain animal cells produce chitin-based cell walls. There are 6 general methods by which living organisms obtain their energy to drive their metabolism and their carbon atoms to build new molecules. These methods are based on the sources of carbon and energy in the environment. Carbon sources include carbon dioxide, methane, and other simple carbon-containing simple molecules, as well as carbon found in organic molecules produced by living organisms, such as lipids and carbohydrates. Sources of energy in the environment that are used by living organisms include light and high potential energy reduced organic and inorganic molecules. Organisms that can synthesize their complex organic compounds using carbon dioxide, methane, or carbon-containing simple molecules are known as autotrophs. Auto means self and troph means feeding. Organisms that rely on organic molecules produced by other living organisms as a source of carbon atoms are known as heterotrophs. Hetero means different. Bacteria and archaea exploit a wide variety of metabolic methods to acquire their energy and source of carbon atoms. All 6 metabolic strategies are found in the domain bacteria. Like bacteria, archaea are multi-strategist, except for the category photoheterotrophism. In the domain eukarya, a large number of species of protists are photosynthetic or photoautotrophs, such as the algae, while the remainder of the lineage is chemoorganoheterotrophs. Fungi and animals are strictly chemoorganoheterotrophs. And finally, all plants are considered as photosynthetic or photoautotrophs. Reproductive strategies in living organisms can be divided into 2 categories: asexual and sexual reproduction. The most common asexual reproductive strategies seen in living organisms are binary fission, budding, and the classical mitosis coupled with cytokinesis. Binary fission involves replication of the genetic material and splitting of the cytoplasm and consequently the cell into 2 nearly equal-sized daughter cells. Binary fission is common in all prokaryotes and some protists such as microalgae. In budding, a new organism develops from an outgrowth or bud due to cell division. The new organism remains attached as it grows, separating from the parent organism only when it is mature. Budding mostly occurs in eukaryotic organisms and can occur in unicellular organisms such as yeast or multicellular organisms including fungi and some animals. Sexual reproduction involves meiosis in the domain eukarya. In the domains archaea and bacteria, meiosis does not take place. Genetic mixing in prokaryotes involves other mechanisms such as conjugation, in which cells replicate certain parts of their genomes and exchange or transfer them to other cells. In the domain eukarya, there exist 3 general types of life cycles. In the first one, the dominant form in which the organism exists is the haploid form. This type of life cycle is known as haplontic and is known to exist in fungi and some fungal-like protists. In this life cycle, meiosis leads to the formation of specialized haploid cells known as spores. Spores divide by mitosis, producing a new organism that is haploid. The second type is known as diplontic, in which the dominant form in which the organism exists is the diploid form. This life cycle is observed mostly in animals. In this life cycle, meiosis results in the formation of gametes. And the last type is known as alternation of generations. In this life cycle, meiosis leads to the formation of haploid spores like in fungi. Spores multiply by mitosis to produce haploid organisms known as gametophytes because they produce gametes. The union of gametes results in the formation of diploid organisms known as sporophytes because they produce spores through the process of meiosis. This type of life cycle is observed in all plants as well as in some protists. When we examine the ecological importance of each of the domains of life, we see that prokaryotes play an important role as decomposers and recyclers of matter in ecosystems. Prokaryotes are the only living organisms that can fix nitrogen from the environment and therefore make nitrogen available to other living organisms. Cyanobacteria are also carbon fixers and use chlorophyll in photosynthesis, and as such contribute to the accumulation of molecular oxygen in the atmosphere. Within the lineage Protista, some are important decomposers, while others play a key role in aquatic food chains where they are key carbon fixers and therefore are the primary source of food and energy for all species in aquatic ecosystems. Photosynthetic protists play a key role in reducing the levels of greenhouse gases and therefore helping reduce global warming. Through their close association with land plants, a group of fungi known as mycorrhizae play a key role in providing nutrients needed for growth by land plants. Fungi are also key decomposers in terrestrial habitats. Plants are extremely valuable as carbon fixers and primary producers both on land and in aquatic environments, thus contributing to the presence of oxygen in the atmosphere. And finally, animals are key consumers both in terrestrial and aquatic environments.